Welcome to Study Live. We are seeing the chapter Biomolecules, and in this session we are studying about the structure of DNA. We know that DNA stands for deoxyribose nucleic acid. This DNA, which is deoxyribose nucleic acid, is the genetic material. Deoxyribose nucleic acid, which is nothing but our DNA. It is the genetic material. It is present. Where it is present? It is present in nucleus. It is present in nucleus of each cell, and it is also present. It is also present in mitochondria and chloroplast. This is in eukaryotes. So in eukaryotes, this DNA is present in the nucleus. It is also present in the mitochondria and chloroplast. Let us see the structure of DNA. When we see DNA, it has a double helical structure. Now it is a double helix which is coiled around a central imaginary axis to form a double helix. So how does this structure gets formed? We are going to see that. So here we can see this is the double helical structure of DNA, where it has got two different strands. So this double helix is coiled around a central imaginary axis to form a double helix. So the other strand we would draw in blue color. So we can see these two strands, they are joined together by hydrogen bonds. So this forms a double helical structure of DNA. Now, when we see this double helical structure, we can see that here it results in the formation of major groove, which is also called as deep grooves, or it is a deep depression. Whereas alternatingly we have over here is minor groove. So it has alternatingly major groove and minor groove. So this distance which is present with one complete turn, with having one minor and one major. So this one complete turn, it is having a measurement of 34 Armstrong unit. And in this 34 Armstrong unit, the distance between two different nucleotides, that distance is 3.4 Armstrongs. This distance is 3.4 Armstrong unit. The diameter of the DNA is 20 Armstrongs. So this is about the double helical structure of DNA. So this DNA, it's a double helical structure which is coiled around a central imaginary axis. Coiled around a central imaginary axis. Central imaginary axis. Now, it is made up of large number of, like when we say chain of DNA, a chain of DNA is made up of large number of units which are called as monomers, and those monomers are nucleotides. This chain of DNA is composed of thousands of repeating units of nucleotides. Repeating units of nucleotides. So let us see this structure and who proposed it. So Watson and Crick have proposed the double helical structure of DNA, and they have proposed Watson and Crick model of DNA in the year 1953 after seeing the X-ray crystallographic studies of Wilkins. So Wilkins and Franklin, they had done experimental studies by using X-ray crystallography, and based upon that Watson and Crick proposed the model of DNA. Watson and Crick in the year 1953 proposed the model of DNA. The model of DNA based on X-ray crystallographic studies of Wilkins, and they were awarded Nobel Prize in 1962. Let us see how this structure of DNA is made. So this structure of DNA is composed of nucleotides. Now let us see this nucleotide. It is composed of, this is called as sugar. So this is sugar. To this sugar we have got nitrogen base attached. Those nitrogen base are purines and pyrimidines. So we have nitrogen base which is getting attached to the first carbon. When they are double ring compounds, they are called as purines. When they are single ring compounds, they are pyrimidines. So as we know, purines include, it can be adenine which stands for A, it can be guanine which stands for G. So this sugar and this sugar and this nitrogen base together form nucleoside. They are forming what? They are forming nucleoside. And when this nucleoside has got phosphate group attached to the fifth carbon, when it has got phosphate group attached to the fifth carbon, so this structure is going to form a nucleotide. So each nucleotide consists of sugar which is attached to nitrogen base at the first carbon by glycosidic bond, and it is attached to the fifth carbon to phosphate group by sugar phosphate bond. So similarly again we have here sugar, to this first carbon attached is a nitrogen base where pyrimidine is there which can be thymine or it can be cytosine, and to the fifth carbon we have got phosphate group. Similarly over here also to this sugar fifth carbon we have got phosphate group. So these are three nucleotides. How are they interconnected? So each nucleotide, so how are they interconnected? The phosphate group of the nucleotide is attached to the third carbon. So this is our third carbon. It is attached by which linkage? This is called as phosphodiester linkage. This is called as phosphodiester linkage. Phosphodiester linkage. So similarly again over here, this is the phosphate group which is attached to the third carbon by which linkage? This is called as phosphodiester linkage. Now we know that DNA is double stranded. This is just one strand. The other strand is going to be anti-parallel. Anti-parallel means it is parallel but one strand runs in upward direction. So the other strand runs in downward direction. So this is our second strand which is running in downward direction. Both are parallel but they are anti-parallel. The two strands of DNA are anti-parallel, which means one strand runs in upward direction, the other strand runs in downward direction. Similarly, we can see over here, we have double ring compound which is purine. So here we will have a single ring compound which is pyrimidine. So here there was a single ring compound pyrimidine. Here we are going to have double ring compound which is called as purine. Double ring compound purine, so we will have a single ring compound pyrimidine. So this is from the nucleoside. To the fifth carbon we will be having phosphate group, which is here we will write 1, 2, 3, 4 and fifth carbon is over here. We have the phosphate group attached 1, 2, 3, 4 and over here we have the fifth carbon which is phosphate group, and over here also fifth carbon we have the phosphate group. Now these nucleotides are attached how? Phosphate group is attached to the third carbon by phosphodiester linkage. They are attached by phosphodiester linkage. Now these nitrogen bases are complementary. These nitrogen bases are complementary, which means purine will always pair with pyrimidine. Let us see about this nucleotides first. Nucleotides are composed of three units which are sugar. This sugar is always deoxyribose sugar, deoxyribose sugar, Then it has got a phosphate group which is attached at the 5th carbon, and there are presence of nitrogen bases. These nitrogen bases can be purines or they can be pyrimidines. They are purines or they are pyrimidines. These purines include adenine and guanine. Purines are adenine and guanine, and pyrimidines are cytosine and thymine. So this was about the nucleotide. Adjacent nucleotides are attached to each other by phosphodiester linkage. So how did we see that? Phosphate group of one nucleotide, phosphate group of one nucleotide is attached to the third carbon, of adjacent nucleotide by phosphodiester linkage. Here we can see this form the nucleotide. Now this pairing of purines and pyrimidines, they are complementary, because when we describe DNA, they are double stranded, they are anti-parallel, and they are complementary. We have already seen what is anti-parallel: one strand running in upward direction, one strand in downward direction. These nitrogen bases are complementary. And this explains a very important rule which is called as Chargaff's rule. One purine always pairs with pyrimidine. And their pairing is highly specific. A always pairs with T by 2 weak hydrogen bonds, and G always pairs with C by 3 weak hydrogen bonds. So here if we have A, so A is going to pair with T and vice versa. Means we have T, so it is going to pair with A. So here we have A, it is going to pair with T by 2 hydrogen bonds. Here we have T, so here it will be A. Between A and T we have got 2 hydrogen bonds. Here we have G, it is going to pair with C by 3 weak hydrogen bonds. So their pairing is highly specific and complementary. Because of that, the ratio of purines, which is A plus G, and ratio of pyrimidines, which is C plus T, is always equal. Let us count over here. We have 1, 2, 3 purines. We have 1, 2, 3 pyrimidines. So their ratio is always going to be 1. This is called as Chargaff's rule, which explains the purines and pyrimidines ratio is always going to be 1:1, and this is explained by Chargaff's rule. Now, when we see this structure of DNA, we can see it has got two ends. This one end, which is having phosphate group, is called as 5' end, because here at the 5th carbon it has a phosphate group, whereas the 3rd carbon over here is free. It is having a free hydroxyl group, so this is called as the 3' end. Similarly, we can see the other end. Over here we have the free hydroxyl group, so this is the 3' end, and here we have the free phosphate group, so it is called as the 5' end. So we can say that these strands are having polarity. Both these strands are having polarity. Both strands have polarity. One strand runs in 5' to 3' end, 5' to 3' end. So other strand runs in 3' to 5' end. Other strand runs in 3' to 5' end, and this is due to, why this is possible? This is due to phosphate group at the 5' end and free hydroxyl group at the 3' end. Phosphate group at 5' end, which is 5' end, and hydroxyl group at the 3' end. hydroxyl group at the 3' end. So because of this we can say both these strands are anti-parallel. So this DNA structure was discovered by and described and proposed by Watson and Crick. So this was Watson and Crick model. With this we complete the structure of DNA, and for more videos please stay tuned with Study Live. Thank you.